Good evening, everybody. My name is Carlos Mora, and I would like to welcome you in the name of Reatch to the second Reatch pitch. And first of all, I would like to explain shortly what Reatch is, for those who have never heard of that. Reatch is, as you may have seen in the logo there also, Reatch stands for Research and Technology in Switzerland. And we are a young think tank consisting out of a bunch of young enthusiastic students. We have been founded 1.5 years ago. We have now more than 65 members already from all fields of science. We are mostly students, graduates, and people that are just interested in science. And what do we want as a think tank? Of course, we want to change the world. And a bit more specific, we want to give science and scientific concepts a stronger voice in the public. Because we think, and you maybe think that too, that in public debates and politics, science really has a weak voice. Sometimes, at least. And as a think tank, on the other hand, we also want to assess new developments in science, and we want to do that critically. And we write blogs about it, we have working groups where we discuss that, and we make events. Also events to inform people about new developments, new hot topic stuff that is coming up. And one of these event formats is the Reatch pitch, the one you're attending. And in the Reatch pitch, we try to short and concisely give you a deep glimpse into a specific field of science, provided by experts in the field, of course, all Reatch members, by the way, and yeah, and I have to look at my notes. Michael, he's PhD, and he's doing his PhD at the University of Groningen in the Netherlands. He has studied interdisciplinary sciences at ETH Zurich with a main focus on biology and chemistry. And he did his PhD about a topic which is really matching that, of course, about controlling biological function with light. And before we start now, I'd like to draw your attention to the little piece of paper in front of you. If you are interested in being kept up or being kept informed about future events of Reatch, about our work, or even want to join Reatch, you can just fill in your name and email address and give me the piece of paper afterwards, or also to Joel here in the back. And you can do that, of course, also on our homepage, www.reatch.ch. And now I have talked enough, and I would like to give the word to Michael and enjoy.
Thank you very much. Hi everyone, good to see you here. And yes, I'm happy to talk today about what I'm actually doing on a daily basis in the lab for the next 45 minutes. So basically what I would like to talk about is how, so 2015 is the Year of the Light, the UNESCO Year of the Light. And as part of that story, I think it's always a good moment to just sit back and think about what actually does light, how does light influence us, also light-based technologies, what kind of influences do they have on us, what influences do we have on light-based technologies, and what is actually happening in that field. And I think it's especially interesting to see what actually is going on in the field of biology, where we now increasingly use light to influence biological systems. And as you will, I hope you will see in the next 45 minutes, is actually that we've been already pretty far, but of course there are many future challenges ahead as well. Yeah, so my point, so basically I'm not really going to talk about my newest result in my PhD, because that's basically already too advanced. I would like to take the time to talk about 4 main basic principles in that field, to give you a general overview, because I feel that not all of you come from a biology or chemistry background, so we'll take our time to go through the most important concepts, that everyone gets the grasp on what is actually going on. And then in the question session afterwards, I'm open to answer any kind of questions. And I also would like to make it pretty interactive, like the whole discussion. So if you feel at any point that you have a question or I explained something not clear enough, just interrupt. Don't throw tomatoes, but maybe raise a hand or shout something. Yeah? Good. Now, yes, so what is it all about? It's basically, the idea is basically to use light to control biological processes. So what we see here is a poor mouse that has an LED implanted in its brain, and basically we can change by switching on the LED or switching off the LED how the mouse behaves. And I would like to explain to you today why that happens. How is it possible that we actually can have an influence on the behavior of organisms, of mice, for example. And this is a purely genetic approach, but I'm a chemist by training, so I would also like you to tell what chemists can do, how they can contribute, and then also what biologists contribute today. And of course, that's a very interdisciplinary topic, so basically everyone that wants to have fun in this topic can contribute for sure. So, and it's also, I would say, an emerging field, so there is still time to actually get involved and do some impact. Okay, so first of all, I would like to start with, I mean, you've all noticed probably life is complicated on a daily basis, of course, also, but even more if you go to the molecular level, if we actually look what is going on, the mechanisms, you know it already, it's extremely complicated and also complex. This is just part of a poster from Roche Metabolic Pathway, where you see all the different enzymes involved in human metabolism and how they interact. And you can see, you can probably spend a whole life to actually understand what's going on. Now, having said that, is what actually, you know, like there in nature there are so many complex or complicated systems out there. And why are we interested in this system? So there are 2 reasons. First of all, by manipulating them or looking at them, we can actually get a closer impression of what is going on. So we can start understanding them, which is of course important for example, developing drugs and stuff like that. But on a second level also it's interesting, just from a conceptual point of view, to actually be able to manipulate the system, to actually not only understand what is going on from here to here, but also to actually start intervening with that system, to tweak here a bit or to tweak here a bit, and through that to actually change how a system behaves. And of course, thinking back about the poster I just showed you, that can become like complex as much as you want. And of course then the main point, if you actually want to influence the system or to manipulate the system, is you have to have tools that actually exert control on different elements of that system. Yeah? So you really have to have tools to actually go in there to change small things in the environment or even single elements of the system that then through the complicated interaction can change completely the outcome of the system. And of course, if you want to control such a system, the tools for the manipulation have to be very specific, and you need to know exactly what you're doing. And that's what the main point of that's the main point of this talk. So natural systems are abundant. They need to be tightly regulated. You can imagine if you have such a bunch of enzymes and you just let them loose, basically that goes all to crap. But fortunately, we're standing here, so something must go okay. And then there are lots of different interactions between the single elements of that system. A good example would be, for example, traffic systems. They can also be pretty complicated, and if something goes wrong, you've all experienced that already, then you have to mess. Now, as I've already introduced, the manipulation of such systems helps us to understand but also to make use of such systems. And 3 important points are there, namely sensitivity, selectivity, and resolution of the control we actually can exert on these systems. And the most important point here in this slide is that the manipulation actually requires stimuli. And what can be stimuli? Stimuli can be molecules, can also be light, and that's what I'm going to talk today. But it could be chemical changes, for example, pH, or mechanical, so by pressing, for example, or whatever you choose. And of course, each different stimulus has its specific characteristics, and by choosing the right stimulus, you also choose, of course, how much you're limited in your approach and in your toolbox. So now, just to bring everyone on the same page, the pharmacological approach is one way to influence complex biological systems. What happens is we take molecules, and I'm sorry, some people that actually study chemistry or biology, that might be a bit of repetition, just really would like to go quickly through it. So what pharmaceutical companies do, they take molecules that specifically interact with enzymes. So it's a molecular approach where we design specific molecules that have a certain bioactivity, and that changes the activity of certain players of this complex system. So we really make molecules that go into the system and change single elements of that system. And you know that's pretty effective. If I have a headache, I go to the pharmacy, get a drug, take it. In most cases, that works pretty well, and my system gets back where I actually wanted it to have. Now, of course, that not always works very well. You know that also yourself. If I take a pill against my headache, I might get problems with my stomach. That can be very unpleasant, so we have the problems of side effects. Also, the drug can have different stabilities. So if you send them, for example, to Africa, let them stand there for 1 year in the sun, they're not going to be happy, and also not going to be active anymore. Then, especially if we talk about bacterial infections, we have the problem of forming resistance. So bacteria get more and more resistant, but not many new antibiotics come up, and that, I'm sure you have heard that in the news. That's already starting to get a huge problem, and the approach using light to actually control drugs might be a solution to that. And also in the same region, of course, drugs always are active in our body, but also outside of our body, they're still active, so they can also exert an environmental impact. That is especially important, for example, in antibiotics, where you can form, as I told you, this resistance, and of course they can also be toxic to the environment, and that's not what we want. So on a very basic level, how do enzymes exert work? Just think about it as some kind of protein that has a part that has a very specific shape. And something extremely important in biology and chemistry as well is that a certain structure results in a certain function. That's a universal, of course, rule. You also see that if you open your toolbox, if you take a screwdriver that has a certain structure, because you need to exert a certain function. And the same applies for every molecule we synthesize in our lab, also every molecule that is flying around in our world. So, a certain molecule, in the case of enzymes, that's what we call a substrate, gets converted through the enzyme to a product. Now, of course, this site here has a very specific shape. So if we take a different molecule with a different shape, that will not fit in there, and will thus not get reacted to the product we actually want. Now, we can make use of that. For example, in the pharmaceutical approach, many drugs actually mimic kind of a substrate we saw here, so they mimic this one, but then no reaction can happen, so you actually kill this enzyme, and that's how pharmaceuticals exert their function. So much to repetition, so that's just an illustration, but that's the inhibitor, and so that's the substrate, this is the inhibitor. The inhibitor can actually kind of prevent the substrate from getting in, we thus prevent a reaction from happening. Good. Now, the idea is, so I told you the pharmaceutical approach is fine. We have nice results. It's a multi-billion-dollar business. But of course, there are some problems. And what we would like to do now is to actually take light to influence normal drugs. So the simple idea is you take light, you shine it on the compound gets active, exerts its function in the body, and then after a while it shuts itself off. That's the whole idea of photopharmacology. And of course you can ask, why would you actually use light? And there are some reasons to use light, namely light is non-invasive. You can control it very precisely because like this laser pointer, you see it's only a single spot. I can decide when I actually turn it on now. I can decide when I switch it off now. And that gives me a huge level of control. Also, photons usually don't, if they're not too energy rich, they actually have quite a lot of, they don't damage any systems. I mean, of course, if you go into holidays, you lie on the beach for a whole day, of course you get sunburned. That's because UV light is more energy rich than actually, for example, red light. So you would not really like to use UV light, but if you use normal, for example, red light, green light, that usually is not toxic to your body, doesn't harm you. So if we can use this kind of light, we can exert control without damaging the body. And that's what we call orthogonal, that basically by using light, we can control processes without interfering with many other processes. Yeah, I mean, of course, if I would like, for example, if I would design a drug that goes into my brain, and I would activate it locally in my brain, but then because I shine light on my brain, also my brain shuts down. That's of course not a good case. So that's what we mean by orthogonality. So our stimulus needs to be orthogonal to actually be able to exert control that only does what we want, but not anything else that has maybe severe side effects. And yeah, also of course I can adjust the intensity of my light spot. And just to sum up, light really is something we can control very well and very sharply and precisely, and that gives us basically lots of room for playing around in combination with our molecules that we design in the lab. So much to that. Now, I told you I would like to talk about 4 different approaches to control molecules using light. Basically, these 4 approaches boil down to 3 ways. And so all these 3 ways, so that's activation, uncaging, and switching, is a way to actually turn on a drug. The only question is, how do we actually turn on the drug? And I would argue from going from left to right, we go up in complexity. So the activation basically is just simple. We start from an inactive drug, we use light that just changes the drug very locally, just as long as we irradiate, it exerts its function, and when we switch it off, also the drug goes back to this unactivated stage. That's so-called photodynamic therapy. That's what I'm going to talk about now. The next part is uncaging, where we actually, it's kind of having, so if my hand is exerting some function, and you can just think I would actually put on a glove that prevents me from doing anything. And basically by shining light on the glove disappears, and they can suddenly start doing some action again. And as you could imagine, this is of course if I have removed my glove, that's an irreversible process. Whereas then the next process, which is called photo switching, is a purely reversible process. And the idea is we have a photo switch that has a certain function. By using light, it changes the structure, and by either waiting or heating or using different wavelength, it goes back. Now, photo switching can be done chemically. That's what I'm doing in a PhD, but it can also be done genetically, and that's called optogenetics. Okay, so let's start with the first part. So first part, the simplest one, is photodynamic therapy. Interestingly, I would say it's also the most developed because it's already in the clinics. You don't have to read all the names here. It's just to show you these are FDA-approved drugs for cancer treatment. So we really see this kind of light activation for drugs is used, has been successful for a few years now, and is really helping to improve lives of cancer patients. So what is the idea? We use so-called molecules that are photosensitizers. What does a photosensitizer do? It's just a molecule. When we start irradiating with light, it changes its molecular organization, and what basically happens in this activated stage is that it changes oxygen. So you know, we are familiar with oxygen, of course, because we breathe that every day to survive, and that's what scientists call triplet oxygen. That's the nice one. But there's also a singlet oxygen. That's just a different state of oxygen, and this is super harmful, super reactive. And if you have singlet oxygen in high concentrations in your cell or like in your body, that immediately leads to cell death. So it destroys cells and thus leads to destruction of tissues. And that's actually what they use. So basically, let's say I have a tumor right here. Hopefully not, but let's just as an example. Now, I administer this photosensitizer. It goes everywhere into the body. But then what they actually do is just irradiate light here, where the cancer actually sits. And just locally, this molecule gets activated. Locally, it transforms the normal oxygen we have everywhere, the triplet oxygen, into singlet oxygen. Singlet oxygen goes wild. It destroys the tissue around it, so all the cells die. And so basically what you do is everywhere you shine light on, you completely destroy the tissue. And that's a pretty harsh method, of course, but it works. And you probably also know that, I mean, cancer is a pretty severe condition. So sometimes you need to take difficult measures to get cured. And that's just basically what they do. They really take lamps. They take red light because that has the most or deepest penetration depth. So basically I can quickly draw that on the board. Simple. What happens is basically if you have your skin, just very schematical, so that's outside, that's inside, so that's the skin. Basically, if you use irradiation, that's at some point going to be absorbed in your skin. Now, if you take UV light, that only goes here, causes local damage to your outer layers of skins. That's why you get sunburned. But if you actually then go to red light or near infrared, so NIR, near infrared, penetration depth of that light is about 2 to 3 centimeters. That's why they use red light there, and that has proven, as I already told you, pretty successful. Now the second point I would like to talk is the so-called uncaging. And that's really something that really works, like uncaging. And how can you imagine how that works? So basically what you have a bioactive molecule that is for most part pretty toxic, and that has some functional groups that are active, that are responsible for this toxicity. That would be, for example, this group here. Now, what chemists do is they actually design certain structures that they just put on this active structure. So basically what happens is, as long as we have this molecule there sitting on the toxic compound, this compound is not toxic at all. So we call it's caged. However, if you then shine light on this, you cleave off this group, and you get to an uncaged compound that is now fully toxic and exerts its function, the function you actually want. And that has been widely applied in biology, especially in the last, let's say, 3 or 4 years. Like the numbers of papers actually reporting that in cellular biology is going up exponentially, I would say. So we basically have every day now, if I go through the ASAPs on journals, we basically have every day now 2 to 3 papers just only about this topic. And the problem, of course, as I already indicated in the beginning, this is an irreversible process. And yes, you generate a toxic compound, but also the thing that you actually cleave off. So if I go back to my analogy of the glove itself is also pretty toxic, and that's a bad thing, of course, because they can also mess around in your system, and that's not something you necessarily want. Okay, this concept has recently been leveled up to something we call selective uncaging. Namely, you can take different molecules that have different gloves. Now let's say glove A gets cleaved off if I use blue light, whereas if I take glove B, the glove B actually gets cleaved off if I use green light. So now I not only have the choice of where and when I remove the glove, but also I can choose by choosing the color of the light I actually use for the irradiation. I can choose which glove I actually remove. Yeah, does that make sense? Okay, so that's what we call wavelength selective uncaging, just schematically depicted here. So basically I have a toxic compound A that is protected, or I have a toxic compound B that is protected. If I shine light, for example green light, on only the B that gets released. If I only shine blue light, then only A gets released. If I shine both get released at the same time. Now, to show you an example that has recently been, last year, been exploited by a group of Deiters and Chen in the United States. So what they use zebrafish as a model organism. The nice thing about zebrafish is that it's actually clear, so you can actually see through them. And of course, that's the ideal case for irradiations. You can also then, of course, see what they actually have changed. That's really nice. So what they do in this case is something they actually, so let's say we fertilize an egg, and then we start growing a zebrafish. And of course, from going from a fertilized egg to the actually final zebrafish, there are quite some steps that have to be successful in his development. And what they now do is they can change the development by just using light. How do they do that? There are certain tools, so-called morpholino oligonucleotides. They have the ability to intervene with certain genes, and the idea is that these morpholino oligonucleotides need to be flexible, otherwise they're not active. So what this group did was taking such a morpholino oligonucleotide, which is the green part here, and they basically just made a circle out of it and put on a glove. Yeah? So as long as the glove is sitting there, it's a circle, and thus not flexible, and it's thus not active. But as soon as I destroy the glove or destroy the protecting group, we open up the structure can now do what it wants, and it gets biologically active. It interferes with a gene metabolism, and thus can exert its biological function. And what they do then, just switch off a gene at an early stage by using one wavelength of irradiation, and then at the later stage they use a different wavelength of irradiation and can then switch off a completely different gene. So by just using two different colors of light, they have the possibility to change two genes in an absolutely normal metabolism in a normal fish by deliberate choice when and how they irradiate. Yeah? And I think that's pretty cool. Okay. Now, the last part, and that's the part I'm working on, it's the so-called photo switching. I would like to first talk about the chemical photo switching and then talk about the biological photo switching. So the chemical photo switching, what is actually going to happen? This is one of the structures we work on a daily basis. I hope everyone had in its high school chemistry that you can have double bonds. It looks like that. And then of course double bonds can either be trans or cis. Very simple concept. Now, what does change if you go from trans to cis? the structure changes. What did I tell you in the beginning of the introduction? Enzymes are very sensitive to changes in structure. Yeah. So because this active site in enzymes has a particular shape, so pharmaceutical companies design molecules that fit exactly into this active pocket. If we now shine light on them and switch from trans to cis, we change the structure. That means then the drug doesn't fit in anymore. Yeah? Or in the more ideal case, first the trans doesn't fit in, but as soon as we irradiate, we change the structure such that it actually fits into the active pocket, and by using light, we thus activate the compound reversibly into an active drug, and then if we want, we can deactivate it again by just using different light. Yeah? Now, this structure here is called an isobenzene. You see this is the trans state. We use UV light to go to the cis state, and then cis is unstable. So if we wait long enough or if we heat strong enough, that goes back automatically. Now you know a human body is about 37 degrees Celsius. It's a little bit warmer than room temperature, and that already means that if we have a drug, switch them on in the body, then after a certain time it deactivates itself. And we can also steer that this deactivation by deliberate choice by, for example, white light or red light. Yeah? So we can go from inactive to active, and active deactivates itself over time, or we can switch it off by choice. Now, exactly. Okay. This field we call photopharmacology. Now, what is important in this field, and I think that's the main argument for this development, is the question, when is this drug active? And of course, if we use conventional drugs, that's always active. So that's active in actually the carton box I buy. It's active before I take it. It's active in my body. It's also active. All the drugs that haven't been used by the body and we excrete are still active in the environment. That can be bad. So that's why they also came up with so-called prodrugs. So these prodrugs are molecules that are inactive, but as soon as you take them, they get changed in the body and start being active. But of course, then they're still active also when they leave the body. Now, if you use light activation, that means that you can only activate the drug we actually need it. And because of the thermal deactivation, it also shuts down itself after a while. So at the time point when it actually gets out there into the environment, then you don't have any problems anymore. Now, a second interesting point that might go a little bit too far, but I think it's nevertheless a very interesting point, is the so-called non-linearity of the dose response. So I quickly can show that again on the board. So if we have, for example, the concentration of a drug and the activity of the drug in the body. Now, like a linear dose response curve would mean that if I use very little, so that's linear, so if I use very little drug, that also exerts quite a small effect on the body. Yeah? So if I want to get a better result, I just have to go up in concentration to go here, and thus get a much better result. Yeah? Now, non-linearity means that, for example, it could be like that. That if I start with a very low concentration, I also don't really change anything in the body, and I can go to a pretty high concentration, I don't have any effect. But if I now go from here to, let's say, here, suddenly you go up like crazy, yeah? So basically, sorry, did I just make a mistake? Yes, sorry, must be like that. My bad, yes. So basically, by changing concentration, you don't change much, but at a certain threshold, you suddenly boost your activity. Yeah? Sorry for that mistake. So basically that means by just having a very small change in structure, in certain ideal cases, we can have a huge effect on the organism. And that's the main point of photopharmacology. So, because if we go back, if you look at the structural change that actually happens here, you could argue, well, actually there's, I mean, of course it looks different, but actually what is happening on a molecular basis is pretty small. Yeah? So the structural change is pretty small. But mostly biological systems are not linear, but they're highly non-linear in their behavior. That means by having a very small change in the structure and thus also in the concentration of the drug, the active drug, we can have a huge effect or a huge change in the result. And that's what all the systems out there usually make use of. Yeah? Now, just to sum up the principle of photopharmacology, we take an inactive drug, doesn't have any effects on the body, doesn't have any side effects, of course, because it's not active, doesn't cause resistance in the case of antibiotics. Now, we take it, in the ideal case, we would start irradiating it, it gets activated very locally, it's active for a certain time that we can choose by designing the molecule, and then gets inactivated after a time, we excrete it, and it's inactivated. And nothing happens, yeah? Here are just two different molecular structures of so-called photoswitches. There are many more, but the azobenzenes, that's this one, or the diaryl ethines are the most famous ones, the ones that are used the most. Okay. Now, if you think about what does a drug for this photopharmacology actually require, that requires, of course, a change in concentration upon irradiation. Then the isomerization should not be only dependent upon UV light, but should be preferentially by using red light. Then the thermal isomerization back to the inactivated one should be not in seconds, but maybe 5 to 10 hours. And we should be able to control it. Then the solubility should be okay. And of course, all the pharmaceutical characteristics should be up to standards on what we use for daily drugs. Yeah? A very nice example, I would argue, is this one. So basically what I took is the ground structure of ciprofloxacin. Ciprofloxacin is a normal antibiotic used in clinics. Currently, I think also in some cases as last resort. Now, they incorporated this photoresponsive element, this azobenzene, and it turns out that this structure here is inactive, and upon irradiating with light, it gets active. And this graph here is just the absorbance of the switch, that's just the way to follow the activity. So that just tells us how much is here, how much is here, and you see basically after time, this is all in cis. After time, we go back to this state. Yeah? Thermal half-life in this case was about 2 hours. Now the nice thing is, and I think that is really cool to show the spatial precision we can achieve, is they take an agar plate, they inoculate it with the bacteria, and they use a mask for irradiation. So certain parts of the Petri dish actually get irradiated, that's this part here, and certain parts don't get irradiated at all. Now, where we actually irradiated, we activate the drug kills the bacteria, and you see here there are no bacteria growing at all, and as soon as where there was no light, actually there are colonies. And I would like to point out, just look at this border, and that's extremely impressive that you can really achieve this kind of control. You can see if I irradiate here on the left side of mine, here I kill everything, but where it's dark on the right side, I don't touch them at all. Yeah. Now, another example is actually from our group some time ago, was they took a membrane channel they incorporated this photo switch into the channel, so into the membrane, into the protein. And what then basically happens is if you shine light on this protein, the photo switch changes, you open up the channel, and you can release cargo. So what they made a vesicle, incorporated this membrane channel, they filled the vesicle with some dye. Now the dye is always in this vesicle, so you don't see it at all. As soon as you actually shine light on, what happens is you open the channel and everything that was inside the vesicle gets out and you suddenly see the dye. And that is currently also used in a related field by a group in Munich, a Turner group, where they try to do vision restoration. So they actually would like to treat blind mice at the moment. So what is the idea? The similar idea is that basically for all your brain's function, we need membrane channels. And the idea is now pretty simple. By using drugs that can block or open the membrane channels, we can steer, of course, how the brain reacts. Now, if you change these drugs into light activatable drugs, then you can change the functioning of your brain by light. And that's basically nothing else than what happens with your eyes, because basically what you have in your eyes are also molecules that are responsive to light. One of the most basic molecules in your eye actually looks pretty similar in the idea to this kind of isobenzene, where you have a double bond that can switch from trans to cis. By this change from trans to cis, you change the structure, and that has some influences then on the nerve cells. They transmit this response, and that changes the brain, and that means also enables you to see. And that's exactly what they wanted to do. So here there's a set of neurotransmitters. So what they do is they take some of these neurotransmitters, they put a photo switch on them, making them photo switchable. And what they basically then do afterwards is they take mouse brains, they take slices of that, put it into physiological buffer, take it into a microscope, check the spiking of the neurons, and as soon as you shine light, you see the neurons starting spiking. That means actually the nerve cells start being active. That's just the depiction of that, so that's what we call an action potential. That just depends on what kind of membrane channels are open or closed, and that's just a neuron. This molecule actually here, that was what I was talking about, that was retinol. You see you have a double bond, namely this one here, that was first trans. If you shine light on it, it goes to cis. And that's actually what is happening in your eyes. Okay. And they use, as I've shown in the first picture, they use these LEDs implanted directly into brains of the mice. And with that, they can change the activity of neurons. And some studies already showed they can actually kind of mimic, for example, dark and brightness of different of your view, for example. Yeah. Yes. To the neurotransmitters. Not that much, no. Because so that's then I think, of course, in your molecular design. That's a good question. You always make the molecular design as such that you don't change the structure too much. Because, of course, if you change the active structure too much, reduce the activity of the compound. So you want to do not change the structure too much, but still make it photoresponsive. So it's a delicate balance in the end then to actually don't interfere with the activity, but then make it photoswitchable. That we can discuss about that also later. And interestingly, we can also change how much and how often a neuron spikes, so I mean how active it is, by changing the color of the light. Because what happens by changing the color of the light, I can go back to here. So basically, if I use, in this case, this is a very simple switch that only switches at 365 nanometers. That's normal UV light. Now if I would go to 450, that means I change the ratio of how much I have here and here. So by changing the wavelength of light, I can also change how much actually is present here or here. And with that, they could also show that they can change the activity of neurons in a certain way, and that also directly relates to this non-linear dose response curves. Yeah. Now, to the last part, before this optogenetics. Before that, I would like to emphasize again that so basically at the moment there are many chemical structures that just operate at UV light. UV light is energy rich, thus pretty easy to actually design switches that operate at that wavelength. The challenge is actually to go to near infrared, and that's also something we do in our labs, to make structures, to come up with chemical structures that are responsive to this light that is less energy rich, and that has been for years now quite a challenge, but also some impressive results from group from Canada, also from different parts of the world. But that's also something that is catching up now. Now, of course, something we could address with photopharmacology is antibiotic resistance, because I can activate a drug that's active for a certain time, but as soon as it leaves the body, it is not active anymore. So we don't have any problem with emergence of resistance in the environment. Then, so that's something here. Also, cancer therapy, that's something we should address in the future, because of course the nice thing about cancers is that they're usually localized. And it's very easy to actually shine light only on a cancerous part, and thus killing it. And I think that's something where this whole field should move to. And it's actually also what you can see now in the recent papers. And the last argument I would like to make for photopharmacology before going to optogenetics is just a very simple one. Chemists have heard that quite a lot. It's so-called chemical space argument. So the idea is basically just if you have carbon atoms, you have hydrogen atoms, you have oxygen atoms, you have nitrogen atoms. Now let's say you take 10 different atoms, you can combine them in any possible way you want. You get a huge possibility of different molecules. Of course, only a subset of this space, of this structural space, is actually going to be biological active. Yeah. Now, let's say this is our chemical space, so that's what's possible. That's what is biological active. And so far, in the last 10 years, we only have used these two switches to actually address the whole space. Of course, not the whole space, but only parts of it, just example-wise. Now, it's very important for the future research that we develop new molecular structures with different functions that allows us to address different parts of the chemical space with different outcomes. And before I actually would like to stop, I would like to talk about optogenetics, and that's a very similar approach to what we have seen in recent restoration. So basically, we also work here with membrane channels. That also changes the activity of neurons. But in contrast to our chemical approach, where we actually design neurotransmitters and make them photoswitchable, we take a genetic engineering approach. We actually take membrane channels that are already photoresponsive and put these genes into other organisms, and that renders them photoresponsive. Now, how do I do with time? Can I quickly show this video? Okay. So this has been, optogenetics has been the Nature Method of the year 2010. So every year a candidate for a Nobel Prize.
Scientists can switch on cells in this mouse's brain simply by switching on a light. The light activates nerve cells, which make the animal walk in circles. This neat trick is also a powerful new tool. Using light to control the behaviour of cells is teaching us about everything from how we wake up to how we learn. This is optogenetics. It all started with algae. In 2002, researchers discovered that a protein that causes green algae to swim towards or away from light is a light-sensitive channel. Blue light causes the channel to open, and positive ions then flood into the cell, much like the influx of ions that cause nerve cells to fire. Neuroscientists were quick to see the potential of these algal proteins. They realised that if they could get proteins called channel rhodopsins to work in mammalian nerve cells, they'd have a very precise way of controlling nerve activity in the brain. For many applications, this method would be better than drugs, which act too slowly, and better than electrical stimulation of the brain, which is less precise. Getting an algal protein into, for example, a mouse brain requires a bit of genetic engineering. First, you take the gene that encodes the protein, plus another piece of DNA called a promoter, and you put them in a virus. When the virus is injected into the mouse's brain, it infects neurons and delivers the gene. A chosen subset of those neurons will have the right machinery to activate the promoter, and in those cells, the algal protein is expressed in the cell membrane. To activate the protein, all you need is light, delivered via a fibre optic cable. It's the ability to target specific neurons in living, moving animals that makes this technique so powerful. And channel rhodopsins are not the only proteins in the Optogenetics toolkit. In this fly, a different light-sensitive system is used to target just 2 out of 200,000 neurons. These neurons govern an escape response, the reflex that makes the fly flee as you move in to swat it. Here, a flash of light activates the neurons, making the fly jump and spread its wings. It can't take off because it's trapped in a petri dish. As well as activating neurons with light, scientists wanted to be able to silence them. Switching neurons off is just a case of using a different light-sensitive protein, such as halorhodopsin, and a different colour of light. Watch how this worm's workout is interrupted when a yellow light is switched on. The light activates halorhodopsin, triggering a flood of negative ions which inhibit the worm's motor neurons and stop it swimming. And neuroscientists aren't the only ones using optogenetics. As well as turning neurons on or off, a diversity of tools allow researchers to control many different processes in living animals and in cells in dishes. These mouse heart cells have been engineered to beat in time to pulses of light. And here, a light-sensitive protein makes this skin cell move towards a laser. The applications are endless. Researchers have already used light to transform the stumbling of rats with Parkinson's disease into a steady walk and show which nerve cells fire when a sleeping mouse wakes. Optogenetics is shining light on everything from animal behaviour to the interactions of proteins inside cells. That's why Nature Methods has selected optogenetics as its Method of the Year 2010.
Good. So basically, what I think you have seen now is that this method is pretty powerful. And it also might be a little bit scary because you can actually do some pretty crazy things. But of course, if you actually would like to use optogenetics for humans, that's going to be a problem because so far we're not allowed to actually genetically engineer humans, and I don't think that's going to happen soon. For, of course, well, for good reasons. Now, that's why I think photopharmacology, so the traditional approach of using drugs to actually interfere with your human functions, but then actually make them responsible to light, might be an option, a viable option to do similar stuff that we can do with optogenetics, but not by genetic engineering, but just by taking a drug. Yeah? Now, and I would like to finish the scientific part of my talk by a quote of Crick, and I think that's what many scientists can just use every day. Is basically the trick is then is not to use the clumsy and inefficient techniques, of classical organic chemistry by themselves, but to make use of nature tools. And the last 2 minutes of my talks, I will quickly talk about my group. So that's the Ferinka group. That's this guy here. We do quite a lot of things. We make molecular motors. We build little nanocars. We use photopharmacology in biology, but we also do material sciences and pure chemistry, where we develop catalysts and new reactions. So that's just what has been done in the past. So they developed a small car with actually 4 wheels, so 4 motors, and put them on a gold surface, and then could actually watch the car moving around up an irradiation of light. And yes, that's where I live at the moment. That's Groningen. That's in the north of the Netherlands. It's about the same height as Hamburg. It's just a student city. Around is not really much. So it's very green and young. So it's quite fun to do research. I actually work in this lab here, so the 8th floor. And as the Netherlands are pretty flat, I can see pretty far, which is nice. In contrast to Switzerland. Yeah, so that's it. Now, are there any questions? Yes.
Okay. If you have such a photo-switchable drug,
Yes.
It's inactive when you excrete it. But I wonder, how big is the chance that it gets reactivated, for example, by sunlight, if it gets to the water circulation? In particular, if you now go towards near infrared.
Yes, that's a very good question. Thank you very much for that. That's indeed the case. So I would have to go into more detail there, but it's a good point. No? So basically, you have 2 regimes. So if we go back to what has been done with the antibiotics. So this drug was purely UV switchable. Now, of course, if you have a UV switchable drug, the problem is that you cannot activate it in your body, because if I have it here and I want to activate it here, and I shine UV light on there, then I have a problem because I just burn myself. But it's not going to activate the drug. So what we need to do in this case is to actually activate the drug. Have a very nice evening. To activate the drug before you actually take it, and because it is active for a certain time, you have a time frame or a window that the drug is active in your body. But then it deactivates itself, and after that, it's quite unlikely that it gets activated again. Depending, because that's of course also a question of concentration of light, how much light you use, like how intense the light is and stuff like that. Now, you're completely right. If you actually then go to near infrared, then of course you have the advantage that you actually can switch on the drug in your body, but then it can also be again switched on later on. So of course then, if it also depends what kind of switches you use for what kind of applications. And of course, you've probably heard, I mean, I'm super enthusiastic about that field, but also because I'm working on in that field. Of course, there are many pitfalls at the moment. There are many challenges ahead, and I think there's a lot of work still needs to be done. Apart from the photodynamic therapy that is now in the clinics, everything else has not been used in the clinics. So, but I mean, that would be, of course, the end goal we would aim for. But on the way to that end goal, there is still quite some work to do. Yeah. And one last thing, actually, which I think is pretty important. So, of course, if you have cancer, and then what we do usually today is you go to the hospital, they actually open up your body, they cut out the cancer, hope they got everything, put it close it again. So then you wait 2 months, and then if you're unlucky, they forgot something. That means you can grow again. They have to maybe reopen you again, and that's unfortunately the fate of many patients. Now, what would be neat, or what has already been, like what people actually do at the moment, they take patients, they open them, they take out whatever they see, and that takes maybe 6 to 8 hours of operation time. But then they take like 3 or 4 liters of a fluorescence like compound. It actually marks the cancer. And then they flush the whole body, like the whole wound with that for some time. Then use light to actually kind of mark the cancerous tissues. And then they see which parts they actually forgot. Now, if you take this even a step further, it would be very nice actually if you would design a photopharma drug that actually gets activated upon light irradiation. That could even be UV light. Now, let's say you actually do this 6-hour operation, but before you close it, you administer this drug locally. And then, by just before actually you close everything, you irradiate with light. Now, of course, as you already know as well, like if you walk around in the sunlight for 1 hour, you don't get sunburn. So it's all about the dosage of the UV light. So if you find the right drug and the right dosage, it should be possible to activate the drug, but you don't cause too much harm to the wound. That would be, for example, an intermediate kind of application where we use UV switches in a smart way. Yeah? So this kind of disadvantages not always need to be a pure disadvantage. Yeah. Further questions? Yes.
So how does these photosensitizers deal with intratumor heterogeneity? Because these drugs would be specific for only a predominant population, and probably the other populations or subpopulations of a tumor are not actually targeted by just the photosensitizers.
Yeah, so I'm not an expert on photosensitizers themselves, but as far as I know, so it's a pretty harsh method. So basically, wherever they are activated, they just kill what is around. So basically, you just make a hole. Yeah.
But that's dependent on the drug that's circulating, because these drugs would be specific for only a certain population, I would assume.
Exactly. So there are different approaches. No. So basically, you could imagine making a drug that is like targeting a specific cancer, and then attach a photosensitizer to it, and then with the hope that you make the killing more specific. Yeah, that would be something like that. Yeah. But I'm actually not 100% sure which drugs are really used in the clinics and how much specific they are. Yes. No, I'm sorry. Is there? Okay. Okay. Okay, then I missed it on my radar. So, but they use light. Yeah. Okay, so not as I know, but there are definitely groups at the moment that really heavily want to go into to neurological like diseases. So using these drugs, and I'm pretty sure in the coming years there will be something coming up.
Are there more questions? So how long do you think will it take until we have kind of a drug in our pharmacy that works with light switches?
That's a good question.
Are we?
That's also a question, of course, we ask ourselves all the time. We always, like, to answer this question, we also have to think about the time frame pharmaceutical companies have to deal with. And the pharmaceutical students among us probably know that it takes quite a lot of time, so at least 10-15 years from the very starting point of a drug to actually the drug getting into the pharmacy. Now, if we now look at this kind of state of research at the moment, I would say at least 20 to 30 years until we really actually have something in the clinics. Of course, that's shelf stable now. But I mean, as we, for example, had in recent case, for example, cholera or something, of course, it's easier to get approval for treatment with experimental drugs if you can prove that the benefit is much higher than actually the risk, because you don't really understand what your company is doing. So if that would be the case in some cases. We might see it earlier, but yeah, that all takes time. But there are also exciting things ahead.
Okay, then I would really like to thank Michael with a huge applause.
Thanks for your attention.
And I wish you a nice evening.